We are always thrilled to have Dr. Lloyd. I think many of you, some of you people who are new may not know Dr. Lloyd. He is a distinguished veterinary PhD with a particular interest in, am I moving your slides by accident? There we go. With a particular interest in, animal models of human disease, and particularly known across our institution for having developed the mouse biology program, where you can essentially order, I'm simplifying it a little bit, you can order a mouse with any human disease you'd like, and Dr. Lloyd can make it for you. We've even had some experience with some obese mice and a few other things that we've done. But it really gives an opportunity, and a great opportunity, to ask important questions about the origin of human diseases in models that you can do in a higher throughput way, and gives us an opportunity to learn about what are the genetic and epigenetic indicators of many of the diseases that we spend so much time taking care of. So it's always a pleasure to have Dr. Lloyd come speak to the department and help educate us on things that are important to the things we think about every day. So Kent, glad to have you again.
So yeah, good morning. It's a pleasure to be here, and thanks for coming again after a 3-day weekend. This is quite a crowd, so it's great. Just a couple disclosures, but neither one of these interactions I have with these groups have any bearing on what I'm going to be talking about today. So when it comes to genotype, phenotype, genetics in general, and how we use genetics, be it in human medicine or in veterinary medicine like myself, I guess this is kind of the holy grail. It's being able to take an associated marker, an associated variant, or some association, and decide whether it's of no consequence, it's benign, it's passenger, or it's causative of disease. And we've now experienced over a decade in which there were genome-wide association studies conducted with the idea is, well, now that we have sequenced the human genome, had subsequent other animal model genomes, why don't we just sequence them all and find commonalities and associate those with disease phenotype? But the trouble with GWAS, is we've come to realize, is that the premise is that diseases are caused by common variations. And that's not an unreasonable assumption in the very beginning, but that's where the problem is, because as it's indicated here, GWAS are looking at such a small portion of the genome, coding sequence in most cases, and the genome isn't that different across populations. As you know, you've always heard things about how we're 97, 98% similar to each other and so on. So the alternative to this has been developed over the years, is that it's really not common variations, but rare variations that have a big impact on disease. Now the plus is that these are quite meaningful for individuals, but the downside is that they're therefore also harder to draw conclusions across populations. So in order to try to find that sweet spot between identifying variants that affect not only individuals, but also applying them across populations, we haven't gotten there yet. So step by step. So here's an example of what it is like to do a GWAS study, or an interpreting in an exome originally, or even now whole genome sequencing, which has become very, very economical to do. So if you look at anyone's genome, there are anywhere from 3 to 5 million different variants that are present. And if you do a general genome analysis, you're going to come up with many, many that are just simple known variants. They're either known because they are known to cause monogenic diseases, they're part of tumor sequencing. They may indicate small effect risks like what you see with 23andMe. It could be pharmacogenetic markers, pharmacogenomic markers to indicate how you will respond or not respond, or metabolize certain drugs, as well as ancestry. But then there's going to be a selection of those that are novel or rare, and they can then follow a spectrum from, and when we talk about monogenic diseases, from either highly pathogenic to benign, so across the entire spectrum. And the ones that are real The real issues here are these VUSs, these variants of unknown significance. And you have the same type of interpretation of tumor sequencing, although they usually divide them up between clinically significant and clinically not significant. So as genomes are sequenced on an annual basis, the number of these variants, these known variants that are associated with these findings here are plateauing. Makes sense. But the real problem is the number of these novel or rare variants, if you look at a graph, are increasing exponentially. Okay? So while that's a lot of data, was known as big data, and now it's being called enormous data, it's good, it's information, but the problem is how to interpret that information. So we are no longer in the realm of either the inability, lacking technology to actually do the sequencing. We are not any longer in the realm of not being able to afford it because it's rather cheap. In fact, I understand there's a place on campus now that could do a whole exome for $100 with interpretation. That's staggering to think about that just of a couple of years ago. But now we're in the realm of how do you interpret that? How do you make sense of that? So a few years ago, NIH's backing the ClinGen, the Clinical Genomic Resource, was assembled with the idea is that why don't we create this central resource to define the clinical relevance of genes and variants for use in medicine and research? And so the idea was put together information from patients, clinicians, laboratories, and researchers that all then, on a very equitable basis, share both genetic and health-related data, and then answer three important questions. One, is the gene itself associated with disease? So like I was mentioning, VUS, a variant of unknown significance, gene would be a gene of unknown significance, a GUS. It's about 21,000 coding genes in the mammalian genome that we look at today. So is this gene associated with the disease? Looking at then, therefore, clinical validity. Is the variant that's seen in that gene associated with disease? And that's pathogenicity. And finally, although the answers to those two questions are important because at least they give a diagnosis, the next step is then, is it actionable? Can something be done about it? Either treating the product of that gene or treating the variant itself and making those changes. It's amazing to think within the last, just comparing the last couple of years, that we can not only ask those questions, but we can now begin to address those questions and do something about it. So then all this goes then into a curated knowledge base in order to improve patient care. So as an example of what ClinGen has done with all this information, is they've classified variants according to this classification. So everything from definitive, these are known variants, either causing disease or not causing disease, so causative or benign, all the way to no evidence reported, and then even these very confusing ones, which are disputed, either because of incomplete health profiles or interpretations that are different, or the genome landscape even changes with time as we get better and better sequencing. So for some of these, the definitive and strong ones, these can be predictive tests that are found in eMERGE, MedSeq, BabySeq, include the moderate ones, and they can be included in diagnostic panels listed here, but all of them give rise to things that can be done for diagnosis and discovery. So there remain, though, big questions with regard to genes of unknown significance and variants of unknown significance. And these are, has the allele been seen before with the disease? And importantly, is it not seen in the normal? It's a good, easy rule out. What effect does it have on the gene product? Does it co-segregate with the phenotype or disease? And what I'm going to try to get into today a little bit is there an in vivo functional and an animal model of the allele, and does it fit with the clinical phenotype? So my thesis that I'm positing with you today is that functional models are critical to determine causation. Again, that holy grail, association causation. And that's even necessary in large patient cohort studies. So I'll ask a question, how large must a patient cohort be to determine whether a disease causation is statistically significant? And the answer is it's got to be pretty big because of all these reasons. There's quite a bit of phenomenological variation in disease symptoms, as you're all aware of. Either VUSs or single nucleotide variants or SNPs, they're not exclusive to disease. 50% may be associated with disease, 12% without disease in some certain studies. You have to account for the zygosity, the dominant recessive nature of the gene product. Statistical significance is also very, very important. So if you think about that, one of the earlier slides, 3 to 4 million variants in all of us, 50,000 variants, you could have at a P05 level, you could see about 50,000 of these being called significant, right? Just out by chance. So therefore, you need a very, very highly stringent statistical analysis. And there's also multiple reasons for association, either linkage disequilibrium, confounding bias, and so on. So a lot of reasons why, despite a large patient group and variations that are common between them, you still need some type of functional analysis. Not all the time, but I think my posit is that you need a lot of time in order to ensure reproducibility. So there are multiple applications for in vivo functional models. Obviously, one of the first is to derive pathogenesis and understanding molecular mechanism of action. It's to create testable platforms, both diagnostic and therapeutic testable platforms in order to predict disease. They're important for being able to assess the role of environmental and other external influences, even behavior and lifestyle. Obviously important for pharmacogenomics. Pharmacogenomics probably being one of the lowest hanging fruit of this entire field, which we should be doing more of, to be able to develop targeted therapies and approaches, and also, very, very importantly, to be able to develop prevention strategies. So what am I doing in this regard, and what is UC Davis? How does UC Davis participate? Well, we participate in a consortium called the International Mouse Phenotype Consortium. And the idea is to build the first comprehensive catalog of mammalian gene function. And there's a number of participants in this. There's 16 sites. You see Davis is here, and 13 countries, and just added Africa to the number of continents on which these laboratories are present. And our goals that we've had for the last 10 years, finishing in 2020, are these. So the idea is to generate a null mouse for every gene in the mammalian genome. Now, when we look at variants in patients, again, be they animal or human, a lot of these variants, they're not causing deletions. They're not functional deletions. They're not causing the type of mutations that are similar to what we see in the mouse. Because when in the mouse, what we do is we take out a critical exon, which is common to all transcripts of that gene, to knock out complete gene function. So you might think, well, wait a minute, that seems rather basic. That doesn't really match a lot of what's seen in patients, and that's true. But that's reflective of where we are today. Remember, I mentioned about 21,000 genes in the mammalian genome. We know the full comprehensive function for maybe about half of those, 2018, and we know the full functional activity of about half. Another half, we know little bits and pieces about, and a sizable number we still don't know any function of them whatsoever. So that's why this project was developed. Sort of along the lines of the Human Genome Sequencing Project was, we have a genome, we all share that, let's sequence it, at least get the basic foundation. The thought of it at that time was that, wow, once we have the human genome sequence, we'll have the cure, knowledge and cure for all diseases in the world. That didn't happen because of the functional adaptation that was needed. Well, in order to get functional adaptation, you have to slog through one at a time and knock out each gene to see what happens. So that's what this consortium is doing. And just like the Human Genome Consortium, which was a worldwide global effort with participation in billions of dollars contributing from many national governments, this one is the same way. So we first make the knockout, and the second step is then we do a comprehensive across-the-board 12-organ system phenotype analysis for each mutant to determine developmental, physiological, molecular, pathological, and biochemical parameters. Then with that, we assemble this baseline data, which is now publicly available for exploring gene function in further depth. And the idea is to develop a profound understanding of the genome landscape itself and the genetic mechanisms of action and what these gene products do. And then also enhance, facilitate interpretation of human genetic data from rare to complex diseases and analysis of large data population sets. Now you think, wow, this is pretty good. Over 10 years to do all this, but this project is only working on the 5% of the genome which is coding, leaving 95% of the genome, which we don't look at in this project. And it's sort of like the universe, right? There's matter is now I'm getting into physics, I don't know, but matter is what, 10% of what we can see, and then there's another 90% of dark matter, dark energy, and so on. So that's kind of the parallel that we have here. And in fact, the data being generated here is beginning to rival the data generated by some of these physics projects, astrophysics projects, and so on. So here's an example of the phenotyping pipelines that we go through. We'll start it here in this top one. This is the embryonic pipeline. And one of the first things we look at is we just look to see, is the knockout that we made, so these are homozygous knockouts, we knock out both alleles, is it viable? Does it live? Does it survive to adulthood? And if it does, follows the green arrow, and we go, okay, the second thing is we say, well, is it fertile? Can it pass on this mutation and still survive? So if it does that, that's great. And then if it survives all that, then it enters the adult pipeline. So I'm going to jump from here down to here just a second. Once we've assumed that this thing is alive and it's fertile, we then grow them up into cohorts, male and female cohorts of 7 and 7, and we enter the pipeline at 9 weeks of age, ending at 15 weeks of age, through a series of tests which analyzes 12 body systems. At the end of 15 weeks, they then enter the 16th week, in which they're sacrificed in tissues, blood, and other samples are taken for doing further analysis. Now, if we back up a second and say, well, what if they're not viable or only partially viable, so subviable? They enter then what's called the embryonic pathway. And the embryonic pathway allows us to do just an essential analysis to see, well, when are they dying during gestation, or when are they dying after birth? And we do a morphological examination, both gross as well as imaging with MRI or with CT, in order to establish a time of death and, as best we can, a cause of death. And this project was just an interesting sideline. When this project was first funded, there was no embryonic phenotyping here, because the idea is that we probably would have a very, very small number of these lines that are lethal or subviable and needing analysis. And it turned out, after the first year, we were finding about 30% of what we were doing could not enter the adult pipeline. That was a huge number. So we all got a little nervous. The NIH and some other governments said, okay, well, let's put some money into this. So we created this embryo pipeline. And now, since 2011 or 2018, 7 years, that number is holding pretty steady. About 29 to 30% of these lines that are taken are embryonic lethal. The other important thing to remember is that we're trying to first focus on genes for which we know nothing about. So not genes that people have already worked on for a long time. There may be models out there. Phenotyping is done, but we're really trying to add new knowledge. We're working on genes for which, if you wrote an R01, you would never get funded, because there's nothing known about it. So our idea is to create new knowledge, understand a little bit about what these genes do in normal biology, see what they do when they're removed, and that then provides fodder and good pilot data for others to take the data and then do, let's say, R01 type research. So this is what we've been doing for about the first 5 or 6 years. It was just a couple of years ago we added then a late adult pipeline. So this is that same pipeline I showed you before at the bottom, expanded a bit. Here's that 9 to 15 weeks again. Because now what we've done is we've taken that cohort and we do it early adult 9 to 16 weeks, and then we allow them to age, because then we also started to see that about 20–30% of these lines aren't seeing anything at the early adult. We thought, well, maybe if we let them age to be more like real adults in terms of humans, we might see something. And we're starting to see, and then we repeat that, the same in-life tests at the late adult stage at about a year to a year and a half, and try to derive phenotype. So that's what we're currently involved in. Okay, so what have we done to date? As of July 2018, we've done, when I say here microinjections, what that means is for every gene we've gone after, we have to either do a microinjection of zygotes or electroporation of zygotes. So we've over 12,000 microinjections. We've genotype confirmed over 7,500 lines, and you've all heard about CRISPR now. So we moved from our embryonic stem cell technology by homologous recombination, embryonic stem cells. When CRISPR came out, we jumped on that, and we were able to now directly apply and improve the CRISPR technique in order to very rapidly generate these. So about 2,000 of these have been done by CRISPR. This is the largest assembly of CRISPR knockouts in animals in one project anywhere in the world. Over 5,800 of these lines have completed the phenotyping pipeline, and public data is publicly available for about 5,500 of these lines, representing over 61 million data points and 370,000 images. So the data is presented both as summary data here, and by clicking on these on the website, you can get down to the individual raw data points, as well as imaging here of both adult and embryos. So here's just, I'm going to give you one example. This is the desmoglein 2 mouse. So desmoglein is a member of the desmoglein, I guess, subfamily of the cadherin superfamily of genes, very important for cell adhesion. It's been known that a mutation, this valine to methionine mutation at 55 position of the protein in this gene has been associated in humans, again, with dilated cardiomyopathy and ventricular rhythm abnormalities. But previous knockout mice for this did not exhibit any cardiac phenotypes. And here's one of the also justifications for this project. As you think, well, wait a minute, people have made knockouts along. But when you do a knockout in your individual lab, let's say the person was doing the DSG2 knockout was a neuroscientist, they're going to look at the brain and throughout the rest of the body, which actually happens. This project, we're totally disease agnostic, and we don't restrict or filter anything. We look at the entire animal. So in knockout mouse made by this project, we actually identified phenotypes involving the heart with dilated cardiomyopathy, increased heart rate, and so on, decreased cardiac muscle contractility, which fit very nicely then the effect of this mutation in the human, which indeed does cause a null. It does cause a complete non-functional gene. It does indeed then confirm what's seen in the human. Okay, so that was just one. And then there's just a list then of several others that have been identified over the last couple of years. So far, and this was a paper from Nature in 2016, where we analyzed this data from the different laboratories and found 360 models of almost 890 Mendelian disease gene associations. And of course, these are the easy ones, relatively easy ones, because there's a Mendelian disease gene association. These are single gene point mutations, single gene knockouts, which in the human cause or an animal cause a known, well-defined syndrome or disease process. So 360 of our models for these then confirm those. The majority, though, are novel, so meaning no previous models found in the literature. And as you can see here, they run the gamut from bone, cardiovascular, neurological, and a lot of them in the eye actually we found. And so since this, we've then had several papers in different, a lot of them in Nature, looking at different disease phenotypes, be they eye, be they developmental, be they hearing screen. So for example, in this hearing one, large-scale hearing loss screen, there were of the 500 genes that were looked at, 300 or so were shown to have a hearing defect. 50 of these were known, but there are another 250 that were completely new and novel. So really, it adds enormous amount of good new knowledge and very interesting information for moving forward. Again, not the be-all and end-all, but at least a way to step forward. So what's next? We're already planning for 2021 to 2030, not that far off. And the idea is now is move from a gene-based analysis to variants, okay? Because that's what we're doing now. And the idea is to do a functional analysis of genetic variation. So with this, our plans are to develop this very, very high throughput, further develop our CRISPR pipeline to deliver phenotyping human coding variants, establish a framework that delivers functional information for these, for now, non-coding sequences. So now we're doing pilot tests now and dipping our toe in the water of the dark genome, the non-coding sequence, especially those enhancer regions, inhibitor regions, and so on, to analyze with these regulatory sequences, how can we mimic the changes that we're seeing in these regulatory sequences that are observed from whole genome sequencing, model those, and be able to inform phenotype. And then also considering the genetic context In order to do a project like this, with global cooperation, we had to select a single genetic background of mouse. For those of you who have any familiarity with mice, in the laboratory world, the B6 mouse is the one that everybody chose, and not just B6, but B6N. So that's also a particular genetic background of inbred mouse. But as you know, we're trying to model the human, which is not inbred. So this is also an issue we have to deal with. With the available resources, we have to be very clever as to how we select which mice to do this. So what we could do is there are multiple categories of mice, depending on their historical origin. And one of the ideas is to select, of the three major categories of mouse populations around the world, is select one for each of these three groups. So at least you get some sense of diversity of the genetic background. And B6 happens to be obviously one of those groups, and we can just select one from each of the other two groups. But also that increases costs, so we have to make some decisions there. And then finally, our idea is to generate clinically informative and diagnostically useful data and reliable and reproducible therapeutic testing platforms. Okay. So our ultimate goal is to really play a central role in functional genomics because it's a lot of data, and so these represent all of the data repositories, well, not all, but the major large mouse data repositories that are available. And then it's also not just about mouse, right? There's also other animals, everything from non-human primates to dogs and cats, the veterinary patients, worms, fish, rats, flies, and so on. So it's important to be able to begin to integrate some of that data into this analysis. And then on this side are just a list of the current human disease analysis projects that we're involved in, such as the Undiagnosed Disease Network, the Center for Mammalian Genomics. Here's the PMI program here, the All of Us, and the corresponding Genomics England program. So we are currently involved in all of these projects right now. And the way to do this is they may come up with, here is a list of variants that we found, or genes first that we found in our patients that are abnormal, or variants that we found. We can't wait any longer. We don't have enough patients showing this, but we have a high suspicion that these are causative. Can you take what we're saying are associated, make a model, and then tell us whether it's causative or not? And so that's currently what we're involved in. So the idea is that we want to, become a gold standard, create gold standard mutants to be very robust and reproducible with sophisticated QC and integrate with human phenotyping data. And our timeline for planning is very short. So this starts here at 2017. I'm not going to go through each of these steps, but the idea is in order to get this into the planning stage, at least at the NIH level, we're working very, very hard right now in order to generate the data, post it publicly, get papers published, and then get it in the timeline for sequencing so that we continue this project after 2021, which is when the current project finishes. So how can we make it all happen here at UC Davis? I'll finish up with this, is that this is an idea I've had now for a little while, and that is to move the idea of animal modeling from one of blue sky discovery to as close to clinical application as possible. So I like to say is how can we make animal models, how can we improve the patient proximity of animal models? Because if you achieve patient proximity, in my mind, you can have a more beneficial impact on people's lives. So the idea behind this is to create a Center for Precision Disease Modeling. And the timing is pretty good for these three reasons. One is there's this growth in translational research as we're all involved in. It's outpaced the development of clinically predictive animal models. The simple knockouts, do a knockout and see what it has, as opposed to here's a human phenotype, let's see if we can model it with an animal. So again, we want to overcome irreproducibility. The second is that there's really only been modest gains in treatment successes and slow and expensive drug pipelines, and due in part to genetic and other omic heterogeneity of populations. So this is this valley of death trying to overcome. And then finally, there's really a lot of breakthrough advances technically in doing genome sequencing and also in manipulating the genome and phenotyping. And this is where we're able to achieve the in vivo functional model in a high throughput, low cost manner. So the key components of a Center for Precision Disease Modeling would be is that it's goal-oriented collaboration between the clinical and research communities, it's to provide centralized resources and services, to establish an evidence-based phenotype and disease ontologies, to conduct clinically relevant and translatable comparative medical research, again, getting to patient proximity, to create in vivo functional models in the most disease-appropriate species, again, getting away from just the mouse, but especially with the advent of CRISPR, which, again, CRISPR is genome agnostic as well. We can now use CRISPR for anything, as you've been obviously reading the scientific as well as the public press. And then also enable the testing and predictive value of new models in preclinical applications. So here's the wiring diagram for this center. It has 3 components. It has a preclinical and co-clinical section. It has a bioinformatics section, and then it has the animal modeling section until it returns straight back to the human setting in order to apply this. So I think we've had the overall general plans of how this would look. It's a matter of generating sufficient enthusiasm here on campus to be able to go forward with this. And I still think we have time to do this, because there aren't too many people around the world that have put something like this together. I think Davis, of any place in the world, I think has the potential and has the resource expertise. Just need the drive and the motivation to pursue this. So what are our goals? Is to derive a functional linkage of genetic disease variant with precise in vivo models, to enhance reproducibility, reliability, and predictive models, to maintain multifaceted research activities, and to create a national, I'd say international resource to bridge the gap between genetic variability, diagnostic accuracy, scientific and clinical knowledge, and this brings in artificial intelligence and machine learning, and ultimately effective, healthier outcomes. And between these can all be linked, but right now there are big gaps between them. So I'll finish up with this, is that Dr. Farmer mentioned mouse biology program, and I think this can grow from mouse biology program, so I just wanted to show you where we were 20 years ago, because this is our 20th anniversary. So in the early days, we were just a small group of people with big ideas. We had made no mice. We had no gene targeting. We had no phenotyping. But we did have a vision, and I think that was important about this, is the vision for a one-stop mouse resource for researchers, staff, and students. And 2018, 20 years later, we're a fully integrated academic program with big ideas. I think we provide a quality product and do quality work with unique, innovative resources, and we have quite an international reputation as an expert in this area. And just last year, we've served 88 UC Davis faculty out of about 115 with NIH grants focused on the mouse, so that's not bad. But more importantly, there are over 828 faculty and researchers served around the world. So this program has a clientele that represents 10% are UC Davis, about 90% are outside UC Davis. We're associated with, on, or at least acknowledge, or these researchers used us in over 100 publications. And the campus really likes seeing this, is we have a positive financial year-end balance, right? So I'm trying to be attuned to that as well. And as I mentioned, we aspire to be a center of excellence in precision disease modeling, and this is some of my group. So that's it. Thank you.
So, Kent, I wasn't too far off when I said you can make a mouse for anything.
Try.
Or trying. We really appreciate that, the great ability to make it understandable for people like us, who most of the time are the end users of all this great scientific knowledge, thinking about, how do we apply, what we see in patients, to how can we roll that back to truly make a difference. I had a couple of questions for you specifically. One was, in your embryology pathway, which you didn't anticipate because there were, what you said, 30% just didn't survive. So makes you wonder, are those the miscarriages that we see, and early miscarriages, things we don't understand? But you were looking at the placentas. Are the placentas different from the mouse? We have an obsession with placentas in my lab.
Absolutely. So we will see embryos that are totally, I wouldn't say normal because they're dying, but we can't identify an actual pathology with them. Whereas the placenta can either have vascular damaged, they're not developing appropriately. So again, because we don't go any further into that, it's again another association. We say, this is a placental defect, causing the embryo death. On the other hand, but that's a small percentage, I would say. I would say of all of the embryo lethality lines, we may see about 10% of those that are placental defects. We'll see most of them, we can clearly see, especially at mid-gestation, probably the number one cause of embryonic death is cardiovascular, because that's about the time, about 12 and a half days gestation, where the heart then takes over the mouse embryo. And you can see a heart defect, and that's usually the cause of death, without any placental defect whatsoever. So with regard to your statement about, end users, so I don't know, you realize that that's no longer an excuse, because here I'm an equine surgeon, right? That's what my training is. And if an equine surgeon could do mouse genetics, so anybody can do this. So, right? So there's no excuse anymore.
Great talk, Dr. Lloyd. Mouse models are obviously a critical resource in research, but there are important differences in the genomics between mice and other mammalians, including humans. Mice have certain genes and humans don't, and vice versa. What is your sense of how, what the prevalence of these differences are, and then how do you think we can better address those important differences in mouse and human genetics?
So very, very good question. First of all, we know with very precise detail what the differences are. Because now with sequencing capabilities, we can directly go in and say, if this is a gene in the human with this particular variant, within seconds, we can go onto a database and say, is this gene homologous in the mouse, and is the site of the variant also a homologous sequence? So far, I'd say the vast majority, because I don't know a percentage, but it's easily more than 90% of genes in the human have homologous orthologous sequences in the mouse. So when we're making these models, we avoid traps in terms of trying to model a phenotype based on a different gene in the mouse that is totally irrelevant when it comes to the human. So that's, I guess, the first answer to the question. The second answer, I'd say, is that, again, going back to the way we can now manipulate the genome using CRISPR technology, we can very, very precisely go in and manipulate the genome of any animal species. So it could be that we see a more relevant gene or variant sequence in a dog or in a pig or in a sheep that we don't see in the mouse, but that is more relevant to the human. So that's the second thing. The third thing is that we're now in a position where we can humanize alleles in any animal species. And of course, the easiest one to do right now is in the mouse. So we can actually take a human sequence, let's say for Alzheimer's, right? We can take ApoE4 mutation. We can take that sequence, pick it up, transpose it right into the mouse, and then, using CRISPR, manipulate that human sequence in the mouse and observe phenotype. So, important question, important concern, but we have many ways of being able to address the small but significant disparities between human and mouse genomes.
It's really profound to think you can go both ways. You can take the human genome piece of interest, put it into a mouse, and see what the effect there is as well. I think Gary first.
So, if I understood you correctly, when you're evaluating these sort of older mice at about 2 weeks or so of age after birth,
We start at 9 weeks.
9 weeks, whatever that
Yep.
number was, right? You're mainly looking at anatomy and not so much physiology, except like with echo, right?
So no, we do both anatomy and physiology. So I went very quickly over just to show you the pipeline, but we'll do a first overall physical exam. Do the whole animal, but then we'll go into a series of physiological tests over the ensuing several weeks. EKG, auditory brain response, grip strength, so musculoskeletal assessment, eye, doing a complete ophthalmological examination. So we go over multiple organ systems, not just anatomy. And at the very end, then we'll do gross, necropsy, and histopathology on those that look like they have something on gross, and then we also select some at random to see if we see something histologic we didn't see on gross. So I would say we cover all the body systems through physiologic analyses. Now, one of the things you may have been picking up on is when I presented that table where I said 440 out of 800 or 900 Mendelian diseases. Well, you think about it, you go, well, wait a minute. If a Mendelian disease is a simple mutation causes a very defined syndrome, and we made mice for 900 of those, and we know we saw 440 that match the symptoms, you might say, well, why is that? You missed them, and that's true. The reason for that is because we can't do every test in the book. So there are some Mendelian diseases that have very defined phenotyping analysis you need to do that we just can't do in our pipeline.
Like cystic fibrosis.
Something like that, that's right. So example, in fact, we do a very poor job, I think, on lung physiology. We did some plethysmographic analyses, a couple of our groups did, but some of this, it has to be very high throughput and keep it in cost. So we're kind of scratching the surface, but doing the best we can. But the idea is that we're taking a gene that, again, as I mentioned, if you try to write an R1 gram, there's nothing known about it, you get discarded. But if we can add a little bit of new knowledge to this, say, there's a suspicion that this causes cardiovascular disease because of this data, that's what we're trying to do. And then people can write a grant on that and do more in-depth phenotyping and then really identify whether it matches a human symptom or not.
Dr. McGee.
I was going to ask about how they do the physical and what they're missing, so
Yeah.
that's a good answer.
Yeah, we're missing a lot, unfortunately, but at the same time, I think we're adding new knowledge that just hasn't been there before.
Mohammed, Dr. Lee.
Kent, always enjoyable. Just putting in a plug for, there's, I guess, a sense that these are just all kind of, genetically engineered, but just putting in a plug for some of the environmental models that you developed and some of the interventional models that's developed, such as the ones obviously that you've done for obesity and bariatric surgery and so forth. And I think that there's some input in that for the group because sometimes for us, finding that great knockout that reproduces a disease, I'll use obesity as an example because that's where my brain is, but you don't necessarily have, you've got a bunch of knockouts that produce obesity and metabolic syndrome, as you know, but not necessarily the same that you would see in humans. So could you, for the group, just address that capability,
Sure.
which produces environmental
Good question.
and interventions?
Yeah, very important question. All of this we do at quote a baseline, right? We're not challenging these mice. We're keeping them happy. They're in a great, they're in a plastic box and the lights for the room and perfect air conditioning. The perfect place a mouse would live. Obviously, that's not true. They want to live in a pile of poop in a chicken farm or something like that. But we keep them in an environment. That's what we have to do. But what Mohammed's alluding to is the fact that we could probably see so much more by challenging them with environmental stimuli. So that's the basis for the late adult pipeline. We're using aging as a challenge, right? It's a challenge for us, it's a challenge for the mice. Another way of doing it is feeding them a high-fat diet. Very, very simple. In some of these, we're going to actually see no phenotype on a normal chow diet, but if we give them a high-fat diet, we then begin to see phenotypic changes. So again, those are the things that we would love to be able to do with the available resources, and we try to do through some pilot tests. The idea is we do the pilot, we see if we get a good hit, and if we do, then we can convince our funders to fund us more. So for example, we just, there's a lot of money coming in for opioid addiction research right now. And so we receive funds. Actually, it was a mandate from Congress that went to different programs. We received funds to be able to identify non-opioid related pathways associated with pain in order to figure out, could we identify some genes producing products gene products that actually could be new potential drug targeting pathways. So that's what we're currently involved in. And we challenge them. We do pain challenges with them. So hopefully that addresses you.
Dr. Greenwald.
Great work. you have any capabilities looking at epigenetic regulators like RNA, or can you link a gene to an epigenetic stimulant?
Yeah, another great question. Again, we don't do that right now. As I mentioned, for the next 10 years, the non-coding sequence was the next avenue, but there are ideas to try to begin to look at epigenetic signaling, because we know that traverses generations and it has huge impact as well. So it's sort of like microbiome. You know, we know it probably has an impact, but based on all the things we've done and how we're trying to keep things standard, it's almost like part of us, we want to keep a blind eye to that because we don't want to upset the apple cart, so to speak. Epigenetics is exactly the same thing, because there's so many different means by which the epigenetics can be modified and changed, and then that having downstream effects on gene expression and function, that it's a daunting task. So to answer your question, we don't do that now, but it is in the thought process moving forward. Great question.
It's great. Well, I just want to say thank you again.
Thank you.